Hey, Sister Mayo, this is our video for lesson 8. I'm doing question 1. Suppose that an individual is heterozygous for the following inversion: the normal chromosome LMN, centromere OPQRSTUVWX, and inversion chromosome LMN, centromere OPQVUTSRWX. Note that alleles, that's not important. Suppose that these two chromosomes replicate prior to meiosis, producing 4 total sister chromatids. The 2 replicated chromosomes then attempt to synapse during prophase 1. The question: what is the allele and centromere composition of the 4 products of meiosis if there's a crossover between S and T? So there's going to be 2 of them that are just like the parent chromosomes. There's going to be one of them that's capital LMN centromere OPQRSTUVWX, and then another one that is the exact chromosome. And then the other 2, one of them is going to be capital LMN centromere OPQRS, and then lowercase TUVQPO centromere NML. So that one's going to have 2 centromeres, and then the other one's going to have no centromeres, and it's going to be capital X WVUT, and then lowercase SRWX.
For the fill in the blanks, we've got non-disjunction in meiosis 2, producing 50% aneuploid gametes, whereas non-disjunction in mitosis can produce a mosaic phenotype. Meiosis in an individual who has suffered a paracentric inversion produces a dicentric chromosome and an acentric chromosome, and an acentric, Turner syndrome is an example of human disease caused by monosomy, while familial Down syndrome is an example of disease caused by Robertsonian translocation. Human liver cells can be 4N8N or 16N. A terminal deletion, chromosome 5, causes Cry-du-Chat syndrome. Crying like a cat. I'm Canadian and French is not my thing. Yeah. While an inversion of the X chromosome causes type A duplication is a change in chromosome structure that can potentially be beneficial to an organism or an entire species. When a translocation causes form, the alternate segregation pattern can produce viable gametes and offspring. So for number 3, these are just questions about defects. And so it's asking about how many total chromosomes would be in these. For Turner, it's 45. With Jacob, it's 47. With the Cry-du-Chat syndrome, it's 46. Familial Down syndrome is 47. And type A is 46. With the Charcot-Marie tooth disease, type 1A, it is not what is not associated would be triploid, aneuploid, reciprocal translocation, trisomy, and non-disjunction. So what is associated would be diploid and duplication. Which terms are associated with Klinefelter syndrome? It would be aneuploid, trisomy, and non-disjunction. And then the following terms associated with type A hemophilia would be none of them. Order.
All right, so for number 4, here is our diagram of what would happen during the synapse phase of prophase 1, and that is just showing which order, and yeah, so that's that. And then for part B of that one, it says identify the allele composition of the gametes produced by alternate segregation. For the first one, for one of the gametes, it was DEFGH.
centromere IJKL, and MNOP centromere QRST. And then the other one was MNOGH centromere IJKL, and then DEFP centromere QRST. Part C, identify the allele composition of the gametes produced by adjacent 1 segregation. For that one, we got DEFGH centromere IJKL with MNOGH centromere IJKL and MNOP centromere QRST and DEFP centromere QRST. And then part D, identify the allele composition of the gametes produced by adjacent 2 segregation. And for that one, we got DEFGH centromere IJKL with DEFP centromere QRST. And for the other one was MNOP centromere QRST with MNOGH centromere IJKL.
Before you go to the next question, I wanted to, we didn't know exactly what you wanted for this diagram, but I think it's worth saying that we understood that for the adjacent, or for the alternate segregation, it was these 2 good chromosomes that they would go into a daughter cell, and these 2 good chromosomes would go into daughter cell, or these 2 translocated chromosomes would go into a daughter cell, but they'd all be viable because they all have the correct alleles, just in different places. And then for adjacent 1, it would be this good one, this normal one with this translocated one, and then this one, this good one with this translocated one, and they would result in viable offspring. And then adjacent 2 would be these 2 going together, and then these 2 going together. We didn't really know how you wanted us to draw that, but I figured explaining it would do the trick.
All right, and then for number 5, it is the cats, saying cats have 38 total chromosomes. How many total chromosomes would be found in the following types of cat? A triploid cat would have a total of 57 chromosomes, as it would be 3N. And so N is 19, and so you times that by 3 and get 57. For B, a cat that is monosomic for chromosome 12 would be 37 chromosomes. Part C, a cat that has suffered a terminal deletion on chromosome 7 would be 38 with a little bit off of 7. And then D, a cat that has suffered a Robertsonian translocation between chromosomes 17 and 18. We decided that it would be either 38 or 39, as there's a possibility of it having one more or the same number. And then part E, a cat that has suffered a reciprocal translocation between chromosomes 6 and 9 would be 38 chromosomes. And that is everything.